All right, let's get started, right? So this is ECEN 689, right? 602, Engineering Applications in Plant Genomics, right? And so we'll be meeting on Tuesdays and Thursdays in this room at this time, okay? And I'm going to try to create uh, a recording of the lecture, right? So that I can give that to you after the class, right? It's worked really well in previous semesters, so I'm just trying to repeat the same thing here, okay? So I have already basically posted this syllabus on the Howdy portal. Okay, but we'll go over it quickly. But if you want some more details, you can go and take a look over there. Right. So this course, I mean, is going to be quite ambitious. Okay, because it's going to be a pretty intense learning experience. Right. However, I mean, how many people here have background in molecular biology? Only one person. Okay, but the good news is that the rest of you also will be fine. Okay, so I'm going to start from level 0, right? And then go from there. Okay, so you have to work really hard. Okay, because for me, believe me, I mean, like, see, 2/3 of this material, okay, is something that I have taught in another course before, right? And I used to cover that in one semester, but it was based on material that I spent 2 years learning. Okay. Now that material is going to be supplemented, okay? Because in the earlier semester, what used to happen was that the focus was more on cancer genomics, right? So the molecular biology portion, organic chemistry review, and all that is the same, okay? But then we used to have paper presentations, right? So this year or this semester, instead of paper presentations, we are going to spend the other third of the semester learning about plant biology, okay, and how it applies, how the methods that we learn here are also going to apply to plant genomics. Okay, so you will see. So to learn that plant genomics portion, it has taken me several months. Okay, I'm still not comfortable. Okay, where I can lecture, but I have another 2 months. Okay, by that time I will be. Okay, I'll have a new set of notes and all that to cover that. So it's really a lot of territory that is going to be covered here, right? And the motivation for the plant genomics thing is that initially we used to work on cancer genomics, right? But A&M, as you know, it's at the Agricultural and Mechanical College of Texas. Okay, so we have a very strong group in the College of Agriculture, and right now we have a joint center between the colleges of engineering and agriculture, right, where the research is focused on plant genomics, right? So in the case of humans, okay, if you're treating cancer, you're trying to cure a disease, right? In the case of plants, you might be trying to increase the yield, right? You might be trying to make plants more drought resistant, right? Or resistant to attack by pathogens, right? But when you actually write down the problem, so you need the domain knowledge to understand what problem you're trying to solve. But then finally, when you maybe write down equations or whatever, or some other kind of model, all right? Yeah, come on in. Yeah, when you write, I've just started, so you haven't really missed anything. So when you write down the models, then the techniques that you're going to use to analyze those systems and to find out what the best kind of intervention is. For example, you might be wanting to, grow plants that are more drought resistant, that have higher yield, and so on. Okay. So how do you do the analysis? So the engineering methods are going to be the same, right? So in this course, my goal is to move beyond the earlier course where we used to focus only on cancer genomics, right? Only the animal portion of it, and to cover the plant genomics also, right? So that you can do research in that area. Okay, and I think given the size of our College of Agriculture, there is no dearth of opportunities and no dearth of problems, right, for people to work on. Okay. So that's the motivation, but in the process, it will be intense because, as I said, I learned something over 2 years, right? And it was hard because it's like a 600-700 page book, right? By the time first time I got towards the end of it, I had forgotten what I had learned before, okay? But the only way I was able to learn it is to teach a class like this, okay? Because when you have to lecture, you have to learn, okay? I cannot come here and say, I don't know anything, okay? You guys are paying your tuition just for nothing, okay? So even for the plant genomics portion also, like I've got the hang of it. Over the summer, I've been reading that. So, and I think in 2 months or so, I will have the notes and all that ready, so that what literally would have taken you like 2-3 years to learn, right, you're going to learn in one semester. In return, what you have to do, you have to keep on studying, okay? Because, it's basically repetitive learning, not memorizing, okay, but you have to look at that thing over and over again. And I will point out similarities with engineering also as we go along, okay? Because the way in which our cells produce energy, it is similar to how electricity is generated, okay? You pump water up to a great height, then let it fall, let it, drive a turbine, right? And then you get hydroelectricity, right? So even in the case of cells, it's similar. Okay, I mean there are protons that are pumped across a membrane to create a gradient, and when things flow down that gradient, you have something that couples it and produces energy. Okay? That's how our cells are powered. So there is a lot of there are lots of similarities all over the place. Like several years ago when I taught the course, I hadn't seen all of those. Because I'm like a newcomer, I just need to get this thing done. But I think this semester we'll be able to do a much better job. Right. So, okay, yeah, so these are the engineering topics like, unearthing intergene relationships, carrying out gene-based classification, modeling genetic regulatory networks, altering their dynamic behavior. These are typically things that we do in engineering, but here it'll be in the context of either animal genomics, all right, or plant genomics, all right, in biology, right. Okay, so, and please don't hesitate to ask questions, if you have any questions, don't hesitate to interrupt me, all right. Uh, Yeah, this stuff you don't need to worry about. Yeah, except the last sentence here, which says that the course should provide training to ensure the long-term success of the TEAS AgriLife Center for Bioinformatics and Genomic Systems Engineering for facilitating collaboration between the colleges of engineering and AgriLife. Because if it is going to be interdisciplinary research, you have to get out of your comfort zone, right? You cannot expect people in the College of Agriculture want to collaborate with us, okay? But we need students who understand what the problems are that they are facing, okay? So, both parties have to get out a little bit out of their comfort zone, and here the main aim of this course is to help you do that. Okay. So if you're going to do research in this area, and these are problems where, the kind of problems that we are focusing on in this course, I don't think they'd get solved anytime soon. Okay. I mean, we'll make progress, but these are not artificial problems, all right? Like, the world's population is growing, okay? So it is a grand challenge to have to feed, a lot more people on the same amount of land, okay, with the same amount of water, okay. So you do need research over there. Or even if you're talking about cancer, okay, people are dying of cancer every day, right? So you make a difference. This is not an artificial problem that somebody created because, let's say, the government dumped in tons of money, okay. So look at it that way, right? Okay, I'm a Datta, and this is my phone number, my email information, and my office location is in the Weisenbaker building. And I'm going to hold office hours immediately after this class, okay, Tuesdays, Thursdays, all right, from 11 to 12. And you can make an appointment, just send me an email if you need to see me, all right. On Tuesdays and Thursdays, I'm planning to be on campus, all right, most of the time, okay, unless I specify otherwise. So you should be able to catch me at other times also, all right, either before the class, after the class, or till like 4 in the afternoon or something like that, okay. Mondays, Wednesdays, Fridays, I'm planning on being at the center, but things will change, all right. Okay, but I just wanted to let you know. And this is the only class I'm teaching, so it's not like you're going to impose on me or I'm too hard pressed. We'll figure out a way. Okay. Now, textbook. All right, the first one is actually a reference for molecular biology, right? It's called Essential Cell Biology, and the edition should be updated because I used to have the 1998 version when I started learning this material, right, about 15 years ago, right, 12, 15 years ago. But so my book walked away, basically. Okay, somebody borrowed it. I never got it back. So I have ordered a brand new copy of this book. I have it with me. It's I think 2015 or something. So you don't have to go and get that book, okay? Because it just so happened that I used to have that book, all right? The second book is very good, all right? It's Plant Physiology and Development. And this is a book that I became aware of when the center was formed. The AgriLife co-director actually said that we should buy and present copies of this book to everybody at the center. That's how I got my copy, and that's that's going to be the source of the material for plant biology that I'll talk about, right? And the problems over there. Okay, now here I listed some articles. Okay, but again, remember, this is kind of new, all right? This plant biology business, we've been working on it only for about maybe 1 to 2 years at most, okay? So we don't have a whole lot of papers. There are some that we will, discuss towards the end of the course, okay? Because I just have like 2 or 3 students that have worked on it. So there are lots and lots of open problems, all right? But so these references, most of them are actually related to cancer, okay? Like you can see, for example, external control in Markovian genetic, that's related to cancer, okay? Or control of stationary behavior in probabilistic Boolean networks. Those are all related to cancer. We may not even discuss all of them, okay? There are more recent ones related to cancer, right? But we'll discuss some of that, but in a very, very condensed form. But 7 and 8, these are references related to plant genomics, okay? And these are the ones that I will probably talk about. Of course, depending on how much time we have, we first have to absorb all the basics, all right? Give the big picture, then we can get into some of the specifics. Are there any questions? Okay, now what all that is fine, okay? But what about evaluation, right? Ultimately, you don't want to just work hard and get lousy grades, right? You don't want that to happen, okay? So what is the grading policy going to be, okay? So there will be 3 exams, all right? Uh, the first one will be on October 4th, all right? Second one on November 3rd, and third one on December 1st, okay? So the first 2 will pretty much be based on molecular biology, right? There won't be any plant biology in it, okay? Stuff related to plants. But the third one is what I've reserved for the plant stuff, okay? And your goal, okay, or what you're responsible for is to learn the material. I'm not asking you to go and do research, right? If you can learn the material and answer the questions in a closed book exam, right? At intervals of 1 month, I'm happy, right? Because your research prowess and all, you'll get a chance. As I said before, there are many open problems. So if you get involved in research in this area, you'll get lots of chances to show your research skills. That is, I mean, it is unreasonable to expect somebody to do research in this class. When in 3 months we have got so much of material to cover, right? But I think at the end of the semester, you will see that, you really learned a lot more, and especially since other than 1%, nobody knows about anything about biology. And don't get discouraged, okay? I didn't do any biology after my 10th grade, okay? So after my 10th grade, then after 23 years to teach this stuff, I had to learn, okay? So you'll be okay. Okay, any questions? So then the listing of the topics I have in my slides, so we'll not worry about it here. And then there is this is just routine stuff, like now lecture notes for the class will be made available to the students. I'm recording my lectures, okay? So I will give you the recordings, right? The MP4 files, all right, so that you can play. And I will also try to see if I can get a PDF file of my lecture also, in case you don't want to spend like 1.5 hours just going through that stuff, okay? Then this is the Americans with Disabilities Act, right? And again, if somebody has, like, see, sometimes for these exams and all that, right, if somebody has a documented disability, right, the university makes arrangements for those people to take the exam separately, right? They are given additional time, right, as appropriate. So, I mean, and again, you don't need to even come and talk to me. If you qualify, then talk to that appropriate office. They will make all the arrangements, right? They contact me before the exam. I have to send it to them. They administer, they return, and all that stuff, right? So that's for your information. And what else? And academic integrity, this is a grad class, okay? I mean, so, and you're going to take in-class exams, okay? So, I mean, what academic dishonesty are you going to do? I mean, you wouldn't be in this class if you were not interested in this material, okay? So, but I have to, just let you know about it, that there are policies in place, there are consequences and all that, but I have so far never had a problem with that, so, okay. And I don't think I'll have it this time. So are there any questions? Okay, now, one more thing I just wanted to mention, like, is it okay if instead of ending the class at 10:50, right? Not today, right? But for other lectures, I ended maybe at 10:55 or at 11, It might change, but if somebody has to walk all the way across campus and all that, we cannot do that, okay? And the reason I'm saying is that if we cover a little bit more material every day, then in the event that due to bad weather or whatever, okay, if I have to miss a class, we don't have to do makeups, all right? And it has worked really great before. But again, I mean, even if there's one person and you're afraid to speak up, okay, you don't want to be the spoiler, send me an email, okay? We won't do it, right? We'll just stick with 9:35 to 10:50. And I cannot extend it too much because in order for the recording to happen properly, I have to stay here for 5 minutes after it's all done, okay, for it to download. So the next instructor will drive me out, So, okay, so let me just get started with the actual course, right? Okay. Now, how do I do? Okay, just, yeah. Okay, now for this portion, I think what I'll have to do is I cannot I'm going to have to use that side. Let's see, whether it messes up anything or not, I did not realize that because I'm not writing, so it's probably better. Did it mess up anything or no? It's still fine? Okay, that's good then. Okay. So let me first provide you with an outline of the course, all right? So first we'll do an introduction, then I'll do a review of organic chemistry, all right? I'm sure you must have had a little bit of organic chemistry before. Even if you didn't, you'll still be fine, okay? I'm going to walk you through the crucial points. Then I'll talk about energy considerations in biochemical reactions, okay? See, many of the reactions that take place in living things, all right, they would not happen even in 10,000 years because there's a big energy barrier that has to be crossed, okay? So I will talk about what makes certain reactions possible, right? Uh, then we will talk about proteins, right? So proteins are very, very important biological molecules. They are responsible for most cellular functions, right? So we'll talk about them in detail. Then we'll talk about DNA. So DNA basically codes for proteins, okay? DNA has all the information that is needed to produce a human being, right? Your DNA has that information to produce you, all right? But that information in the DNA is first written in a slightly different language, which is called RNA, and we'll talk about that, right? And the process is called transcription. And then the code in the DNA is basically interpreted or translated to produce proteins, right? Then we'll talk about the organization of the DNA in cells, right? And DNA is organized in the form of chromosomes. Then we'll talk about how, genes or a stretch of DNA that codes for a protein, right? What decides whether a particular protein is going to be produced or not, okay? That's called gene regulation. Then we'll talk about genetic variation because, is this kind of genetic variation the survival of the fittest, okay? That has given rise to the organisms that we see around us today, okay, over, evolutionary time. Now that is the kind of variation that occurs, uh, very randomly, but in nature it occurs. Like if you're trying to kill a bacterium, okay, we're using antibiotics, right? Most of the bacteria will die, okay, but one or two of them will survive and they'll take over the population. So you do have this kind of genetic variation, but this kind of variation arises randomly in nature, okay. Today you have the technology. This is DNA technology where you can artificially make changes in the DNA, right, and alter the DNA and then put it back into the organism and you get mutant organisms, right. So that is DNA technology. Then we'll talk about cell division because that is the basis of all life, okay. The only way to create new life is by division of existing cells, okay. So we'll talk about cell division. That's very important. And then we'll talk about cell cycle control, cell death, and again here, if it was the other class, it would have been cancer, but here we'll talk about plant senescence. That means plants growing old. They go through a cycle, right? I mean, in the spring everything blooms, and then in the fall leaves start falling off, and so on. Okay, so then these are all topics in molecular biology, right? And a little bit of plant biology too. But then you basically want to measure signals, all right? So these are because if you're going to use engineering, you have to do some measurements, all right? So you're going to measure the activity of genes, okay? So genes code for proteins. You're going to measure the activity of genes using what are called expression microarrays. Again, I'll explain all of that in detail later, right? This is like a gene chip where you measure the activity of thousands of genes at the same time, or using a technique that is called next generation sequencing, all right? First, you have to know what is the earlier generation sequencing. I will cover that. Okay. Then we talk about next generation sequencing. So that step, the first bullet on this page, that is going to give you the kind of data that you need for applying the engineering methods. Then you apply the engineering methods and you might do disease classification, right? For example, you got data from some cancer tissue, right? And you want to classify the stage of the cancer, okay? So you have to do, and that is a standard engineering thing, okay? If you look in the area of pattern recognition, machine learning, right? They cover these things. So classification, and these are some of the methods that are used. K nearest neighbor, LDA is linear discriminant analysis, QDA is quadratic discriminant analysis, and perceptrons. I will cover those things in detail. It'll be much easier for me to cover those because those are engineering topics, okay? And I'm basically teaching an engineering audience here, right? Then I will cover clustering, right? Clustering means if you see, if you see a bunch of genes, okay, you want to group them together, okay? You want to discover patterns, okay? You want to find, so clustering has also uses different techniques. And again, these are standard methods, okay? Like what we are not going to do in this class is, I'm not going to make this into a class on classification, all right? or on clustering, okay, because it is not, okay. There is a course on pattern recognition that you can take for that, right? So here the focus will be on building up the domain knowledge and doing like simple classification, simple clustering, all right, just to see how things map together and to get the big picture. Because you're engineers, you can run with clustering and classification. I don't need to teach you that in this class, all right. Then we will also do Markovian modeling of genetic regulatory networks, okay, how the genes interact with each other, okay. And it is a Markovian system because, see, think of it this way, all right. Given the information about your parents, okay, I don't need to know anything about your ancestors, okay, to make predictions about you, okay. So it is Markovian. It's a one-step Markovian thing, right? So these genetic regulatory networks also, we will look at them as Markovian genetic regulatory networks, all right, where the state evolves like a dynamical system, all right, and how you could control them to make the system go towards the desirable state. Now, whether it is Markovian or not, I don't know. Whether it is one-step Markovian or not, all right, we don't know. Only the results will tell us, okay. If you come up with some results based on a Markovian assumption and your data checks out and things work out, right, then you say it is Markovian, okay, because our goal, I mean, we don't know whether it is Markovian or not, okay, but I'm saying that intuitively because, as I said, in biology, right, given, let's say, your immediate parents, I don't need the information about your ancestors, so it's only a one-step thing, right, and many of you, how many of you have taken Professor Kumar's class on stochastic systems, right? You don't need this for this class, okay? But he talks about all these Markov chains and Markovian systems, right? But I think in biology, there is that model is appropriate for biology. It's just that we do not know all the parameters and things like that. Learning those is going to be a different story. And then after that, I will focus on applications in plant water use, plant breeding, and precision agriculture. Okay, that's what I mentioned, right? And again, this is a weekly listing, all right, of the syllabus, all right, what is going to be covered each week, all right? But we will deviate from that because that last topic, as I said, I'll spend 1 month covering that, okay? I'm optimistic that I can finish those other things in a couple of months, and then 1 month I'll spend on this because this is a course on engineering applications in plant genomics. All right, I want you to leave this class with sufficient knowledge about plant genomics, all right, to see what problems are important, all right, so that if you want to do research, you can just jump in. All right. Okay, any questions? Okay, so let's now start with an introduction, right? So if you're going to do molecular biology, you have to talk about cells. A cell is the basic unit of life, right? Nothing smaller than a cell can be called living. Okay, for example, a virus, right? Virus is not a living object. Okay, although it makes you and me sick, right? Virus is not a living object. A virus is something uh, that can make multiple copies of itself, right? By exploiting the machinery of the cell. Okay, and then it kills the cell in the process. Right? So virus is not a living object, but the cell is the basic unit of life, and this thing. It got its name based on the work of Robert Hooke, okay, who was the first one that used this term because he examined a piece of cork under a microscope and he basically saw the dead cell walls. Okay, so he called it a cell, like a jail cell, right? So that's how the word cell came into being, right? Now all living creatures are made of cells, and typically, I mean, again, we are saying 5 to 20 micrometers in diameter, but the size range is huge. Okay, I mean, if you're looking at a frog's egg, it's about 1 millimeter in diameter. That's a pretty big cell, right? If you're looking at the bacteria that live in, let's say, yogurt, lactobacillus bacteria, right? That's really, really small, okay, in micrometers, okay. So there's a large variety, right? Now, if you look at a typical cell, and I'm picking a plant cell here because this course is going to be about plant genomics, all right? So you have a delimiting membrane, all right, which is the plasma membrane. That's the membrane that is outside here, all right? So you have a delimiting membrane that is called the plasma membrane, right? That contains, that's the outer boundary of the cell. And inside the cell, you have several compartments, right? Like you have the nucleus, which contains the DNA, right, of the cell. Then you have the mitochondrion, okay, which is the site where energy generation takes place, all right? Then you have the chloroplast, because it's a plant cell, it'll have chloroplasts, all right? Chloroplast is where photosynthesis takes place, all right? Then you have lots of organelles. We've just picked up some of them. You have an organelle that is called a peroxisome at the bottom of the picture, right? That's the organelle where reactions which generate hydrogen peroxide take place, okay? Because hydrogen peroxide is toxic for the cell also, right? Then you have the boundary of the nucleus, which is called the endoplasmic reticulum, which is where a lot of the cellular components are synthesized, right? See, each cell is like a factory, okay? Although despite its small size, despite its diminutive size, okay, it is like a factory where thousands of reactions take place each second, right? So the endoplasmic reticulum is where some of the materials needed by the cell are produced, okay? And then the Golgi apparatus is the place where they are modified, okay? All of this stuff is actually captured on the next slide, okay, where this information is written down, right? So mitochondria is energy generation, chloroplast is the site where photosynthesis takes place. The nucleus contains most of the DNA of the cell, right? The mitochondria and the chloroplast also contain a little bit of DNA, but most of it is in the nucleus, right? Now, if you take a cell and you remove all those internal organelles, right, then what remains is like a soup, watery soup, right? And that's called the cytosol, okay? So it's a cell stripped of all the organelles, right? So you have a plasma membrane, you have a cytosol, and you have all these organelles that are sitting inside, okay? And again, anytime you have a question on any particular slide, just stop me, okay? I'll be more than happy to elaborate. Provided, of course, I know the answer. Probably I will, because you're seeing it the first time. You know? Now, if you're looking at cells, there are two broad categories of cells, right? Prokaryotes, right? Or prokaryotic cells, they do not contain a nucleus and other organelles, right? So that means this is the class of cells that don't have any of those organelles inside. Okay, bacteria don't have that. Okay, so they are called prokaryotes. Pro means before, and karyon is a nucleus or a kernel, right? And the other category of cells, these are what are called eukaryotes, right? They have a nucleus, right? And they'll have other organelles too, right? And humans are eukaryotes, right? Human cells are eukaryotic cells, yeast cells, all right? So you have prokaryotes, synonymous with bacteria, right? And eukaryotes would be humans and everything else, okay? And there are other categories, but let's not worry about that, okay? There's one more category, but we'll not worry about that here. Okay. So if you look in the cell, as I pointed out, right, the cytosol is that watery gel that you have when you take out all the organelles from the cell, okay? Now the membrane, the plasma membrane, the delimiting membrane, right? See, that is not watery. You know that, I mean, even from your childhood experience, that oil and water do not mix, right? So the inside of the cell, that is watery. That's an aqueous environment, right? The membranes are all made up of lipids. It's like oil, okay? So they will stay away from each other. That's how the cell is structured, right? So the characteristic that these lipids have, right? And I will point out when I do the chapter on review of organic chemistry, I will point out why they have this characteristic. But lipids have a part, a lipid molecule has a part which is hydrophilic. That means it loves water. And there is a part that is hydrophobic, that hates water, okay? So because of that, lipids are able to produce cell membranes, okay? And the cell membrane is typically a lipid bilayer, as I'm going to explain in the next couple of slides. Any questions so far? Okay, so what do you see at the top over there? Okay, that molecule, that single molecule, it has got a hydrophilic head, right? That is the black dot over there, right? And then it has got a hydrophobic tail, right? Right. Actually, it has got two hydrophobic parts, and I will explain to you later, okay, when we cover the review of organic chemistry, why that is the case, right? So a molecule like this has got a portion that black dot, right? That really likes water, right? Another portion that hates water. So guess what? When you throw such molecules in water, what are they going to do? They are going to line up so that the portion that loves water faces the water, the portion that hates water stays away from the water. Okay, and that's how this membrane here is going to be formed, right? Okay, so you can see that on the left, right? You can see that there is water inside, right? Water outside, right? And the portion and these molecules, they have lined up in a way where the portion that likes water is facing the water, the portion that hates water is staying away from the water, right? And this is called a lipid bilayer because there are two layers, and this is how cell membranes are formed. Now this membrane, it has got really uh, amazing characteristics. Like if you take a pin, okay, and pierce that membrane, okay, things will move apart, the pin will go through, right? You remove the pin, everything will reform, okay? Because the portion that hates water will continue to hate water, the portion that loves water will continue to love water, right? So that's one aspect. The other aspect, and the reason why even in this introductory chapter I'm covering this in detail, when I'm talking about this in detail, is because this is not a topic that we will consider in detail later on. Okay, we don't have individual. I mean, if you look at the first molecular biology reference, that has a chapter devoted to it. Okay, but I'm trying to give you the big picture, the gist of it, without going into all the details because we have too much of material to cover. Now, the other thing is that the lipid bilayer also allows what is called vesicular transport, right? Because suppose you have some aqueous cargo, okay, that you want to move into the cell, okay? So what can you do? You can package that aqueous cargo, all right, inside a lipid environment. You can encapsulate it inside a lipid, okay? Bring it close to the cell membrane, okay? Then the cell membrane is a lipid, the membrane of this one is also a lipid. The two will fuse, right? Then the aqueous cargo can go inside. Okay. Same way, if you want to transport something from the cell membrane, right, to the interior, one of the other organelles of the cell. Okay. Again, so from the cell membrane, you can take some lipid. It can encapsulate the aqueous cargo that you want to transport, right? And that's called a vesicle, right? That thing over there, that circular thing is called a vesicle, right? So it's taken some content from outside, right? Encapsulated inside the cell membrane. Then it'll travel, right? And then again the membrane will fuse. Once it reaches its destination, the two membranes will fuse and the aqueous cargo is going to get transferred. Okay. And again, I mean, there is an elaborate mailing system. Don't think that only UPS and the US Post Office, they know how to do mailing. Okay, the cells have been doing it for millions of years. Okay, like they will tag a cargo that is destined for a particular location, right, differently from cargo that is destined for some other location, right? Okay, any questions? Now, another thing that I want to talk about in the case of cells is the cytoskeleton and its functions. Okay, so as you can make out, cyto means cell. Okay, cytoskeleton means the skeleton of a cell. Okay, now if you look at the interior of a cell, I said the membrane is made up of lipids. You have those organelles. Okay, but if you look at the cytosol, it is not a structureless soup. Okay, it is an aqueous soup. All right, but there is structure to it. All right, so there is reinforcement inside. So there's something like a skeleton. Okay, and indeed, you know, there is a chapter in the book, in the molecular biology book, that is devoted to the organization of the cytoskeleton, right? And uh, like I have just picked up the important things from there. There are three kinds of molecules that make up the cytoskeleton. The first one is called actin filaments, okay, and these are all proteins. These actin filaments, they will generate the contractile forces. Example, in muscle cells. That's how you and I move. Okay, all the muscles together, they have to, the actin filaments have to generate the contractile forces. It's across the entire set of cells or the tissue, and that's how we move. Okay, we will talk about that. How you get the energy for doing that. Then another set of proteins, right, that form part of the cytoskeleton. These are called microtubules, and they provide tracks along which different cell components can be moved. Remember, I said that inside the cell you can transport from one organelle to another. Okay, so you would need some tracks. Okay, some highways. Okay, network of highways. But in the case of the cell, the difference is that the highways are laid out on an as-needed basis. Okay, you don't have the highway connecting, let's say, College Station to Houston, right? Laid out all the time. If I need to go there, then that thing will be laid out and I go. Okay, and then again, it's taken apart after that. Okay, that's how it works. All right. So microtubules provide tracks along which different cell components can be moved. And another very important role that they play is that they distribute the DNA properly between the daughter cells at cell division. Remember, when the cell divides, okay, everything is going to get doubled, okay, because you're going to get two cells instead of one. The DNA has to be replicated, all right? And I mean, we'll get to the structure of DNA later, but you have like 3 billion letters, okay? They have to be replicated with as few errors as possible, and then properly partition between the two cells at cell division. Okay, so that requires some kind of machinery. Okay, and the microtubules can form something that is called the mitotic spindle that is going to do that. Okay. Another class of proteins are what are called intermediate filaments. These provide mechanical strength to the cell and its neighbors. Okay, because otherwise the cell might collapse. Okay, right. So for some things you need a certain amount of rigidity. Okay, and that's provided by intermediate filaments. Now, we have said, cells vary a lot. Like we said, the size can be really small, it can be really big, and cells look different. The cells that make up your bones look different from the cells that make up your hair and so on, okay? Or even across different organisms, the cells look different, okay? However, the good news is that all cells have the same underlying chemistry, right? And the genetic information in the DNA is coded using the same blocks in all organisms. So if you take DNA from humans or from mice or from ants or whatever, the basic modules are the same. Okay, so that's why it makes it much easier for us to study that, and especially for engineers. Otherwise, we'd have to memorize a lot. Okay, we don't have to memorize that much. Not only that, this information also is interpreted using essentially the same machinery to produce proteins. Okay, the same code is used. All right, and in fact, proteins are made up of the same 20 blocks. Okay, no matter which organism you look at, whether it's the lowest form of life, all right, or human, all proteins are made up of the same 20 blocks that are combined together, right, in different lengths and in different patterns, right. As for, and here I like to quote one of my colleagues. Like when he gives talks and all, he'll talk about those 20 amino acids on this planet. Okay, if life exists on another planet, we don't know. Maybe there are other amino acids that are used. If amino acids are indeed the building blocks of those proteins. So this greatly simplifies the study of many aspects of molecular biology. It does not have to be organism specific. That's why I said, like whatever I had taught in those earlier courses in the context of cancer and animal biology, that's still applicable right now, right? I don't need to start from scratch, right? Those notes can be used because it's the same structure. And there's some subtle differences, but other than that, it's the same. Any questions? Okay, now if you're going to do molecular biology studies, all right, you need model organisms, okay? Because every time you want to study a phenomenon, you need some organisms that will produce a lot of descendants within a short period of time, okay? You cannot wait for eternity for, for an organism to mature before you can carry out studies, okay? So model organisms are organisms that are used for molecular biology studies. They have to be simple and capable of quickly replicating themselves, right? So if you're studying bacteria, the model organism of choice is E. coli, right? They contain only a few genes, right? Genes means basically DNA that codes for proteins, right? And they reproduce every 20 minutes, okay? So you can see that if they reproduce every 20 minutes, okay, if I start with one bacterium, okay, at the end of 1 hour, how many bacteria am I going to have? Let's say all the bacteria that are produced, not 6, 2 to the power of 3, yeah. So I'll have 8 bacteria, yeah. So you can calculate in 24 hours, you'll have more than 1 billion, okay. So maybe in a couple of days, if you let E. coli multiply, there will be more members than there are humans on this earth, okay. That's how quickly they multiply, yeah. So it's easy, like if you, for example, if you're going to look at genetic variation, right? Like I said, that when, over evolutionary time, right, is the survival of the fittest, right? So with bacteria, you can demonstrate that in the lab because, over, in the course of 24 hours, you have more than 1 billion, right? So what you can do is, so they are going to multiply. So you treat with antibiotics, all right? And if you let it continue for a day or two, most of the bacteria are killed. But the ones that survive, they will take over the population because they're going to multiply like 1 billion times every 24 hours, right? So you can see genetic variation at work because the ones that survive, they are the ones that probably have some difference in their DNA. Okay, that is allowing them to evade the effect of the antibiotic. Right? We'll talk about that later. Yeah. Huh? What? No, no, no. They have DNA, but it's not inside a nucleus. No, no. Bacteria have DNA. Yeah. So for flowering plants, it is the plant which is called Arabidopsis, right? And that produces thousands of offsprings in 8 to 10 weeks, right? Now, if you're looking at insects, it's the Drosophila or the fruit fly, right? And for mammals, it's rats and mice. Okay, obviously it cannot be humans, right? I mean, it cannot be model organisms. And for unicellular eukaryotes, right? That means unicellular single cell organisms, right? But that have a nucleus, it is the yeast. Okay, any questions on this introductory chapter? This was just to get you started, and I will get into the review of organic chemistry now. Any questions on this? So again, I mean, even for eukaryotic cells, okay, there is DNA also in the mitochondria and in the chloroplast. Okay, there is DNA, but the bulk of the DNA is actually in the nucleus. In fact, when you inherit, right, traits from you inherit traits from both your parents, all right, through the DNA, okay. But from your mom, you inherit more because you get stuff from the some mitochondrial DNA also. Okay. Okay, so let's get started with the review of organic chemistry, right? And this stuff is going to look really, really easy when I get started because everybody knows everything, okay? But by the time I get towards the end, okay, I'm sure everybody will find something that they didn't know, and then they will have an aha moment, okay? Oh, now I see the connection, okay? Because because I will start with elements and atoms and things like that. Everybody knows that, okay? So what is an element, right? So you cannot break it down or convert it into another substance by chemical means. Atom is the smallest particle of an element that still retains its distinctive chemical properties, right? Then molecules are formed by two or more atoms of the same element or of different elements combining together. No questions there. Okay, everybody is familiar with that. And again, if somebody is not, if you require more explanation, let me know. But I'm saying that everybody has done high school, chemistry, and so you know this stuff. But how many of you know about, the fact that lipids have a hydrophilic part and a hydrophobic part? So one person, yeah, two of you know. The others also, I'll show you why that is the case. Okay. By the time we get through all this, and we'll see that. So inside an atom also you will have these subatomic particles, protons, right, which have mass of 1, charge of 1, right, positively charged. Neutrons, mass 1, charge 0, no charge. Electrons, mass 0 and charge -1. Okay. And again, if you're a nuclear physicist, you probably will be able to dig in a lot of holes into whatever I'm saying here. That is not the intent. Okay. Here the intent is to cover the minimum background, right, to explain the biological stuff, right. Intent is not to come up with the most accurate physics and say, okay, yeah, no, these definitions are wrong, and there are smaller particles that have been discovered and all that. That's happening all the time, but that's not the goal in this class. Okay. So the protons and neutrons reside inside the nucleus, and the electrons revolve around the nucleus in certain orbits, right? So if you look at an element, right, it'll have an atomic number, which is the number of protons in the nucleus of an atom, right? And here are some examples. And again, the examples that have been chosen here are the elements which are relevant in biology, right? So hydrogen has an atomic number of 1, oxygen has an atomic number of 8, carbon has an atomic number of 6, nitrogen has an atomic number of 7, phosphorus has an atomic number of 15, sodium has an atomic number of 11, calcium has an atomic number of 20. Okay. Why did we pick these? Again, because these are the elements that occur over and over again in biological molecules, right? What is the atomic weight? Well, the atomic weight is the total number of protons and neutrons in the nucleus of an atom, right? And it is also equal to the mass of an atom relative to that of a hydrogen atom. Because a hydrogen atom has got only one proton, one electron. Electron doesn't contribute to the weight, so the atomic weight of hydrogen is 1. Okay. So since the atom is electrically neutral, right, number of positive charges must be equal to the number of negative charges. So number of protons must be equal to the number of electrons. The number of neutrons in an atom of a particular element can vary, right? And that's where you get isotopes, right? For example, carbon usually will have 6 protons and 6 neutrons. So the normal atomic weight of carbon is 12, but carbon atoms possessing 6 protons and 8 neutrons also exist, right, where the atomic weight is 14, right? And so when you have atoms of the same element with varying number of neutrons, they're called isotopes. And usually one of the isotopes is stable, is chemically stable, right? While if you start with any of the other isotopes, then it is going to radioactively decay to the other one. Okay, and you calculate. In physics, you've done all those things like calculation of half-life and things like that. Okay. And in fact, this carbon with atomic weight 14, okay, that is the basis of this technique that is called carbon dating, because you can see by how much it has decayed, right? And you can figure out how old that fossil is, right? So the protons and neutrons are held together tightly in the nucleus, while the electrons revolve around the nucleus in certain discrete orbits which are called shells, right? So this is a typical schematic diagram of an atom. So in the center you have the protons and neutrons inside the nucleus of the atom, and then around it you have these shells. Okay. Now the number of electrons that can be accommodated in each of these shells, right? It's given by a formula, right? The first shell can hold up to 2 electrons, right? And then it's completely filled. then the second shell can hold up to 8 electrons, the third shell can hold up to 8, and so on. Okay, there is a formula that can be used to calculate these things, right? And again, I mean, this is not an exhaustive course on chemistry, so I'm just going to, not go into too much of detail about that. Okay, I'll just cover whatever I need. So if you look at the inert gases, they have completely filled outermost shells. Like if you look at helium, it has got an atomic number of 2. That means it has got only one electronic shell, and that's completely filled up. So it does not react with anything, right? If you look at neon, right, that's the next inert gas. It has an atomic number of 10, which means that the electronic distribution will be 2 in the first shell and then 8 in the next one. Outermost shell is completely filled, so the atom is unreactive, right? If you look at argon, right, atomic number is 18. Again, it's 2 plus 8, 10, plus 8, 18. Outermost shell is completely filled. These are all inert gases. Okay. So next we start looking at elements that actually react, okay, which don't have completely filled outermost shells. An example would be sodium, right, which has got an atomic number of 11, right? So that means it has got 11 protons in its nucleus, right? So it has to have 11 electrons. So the electronic distribution is going to be 2 plus 8 plus 1, okay? So it has got an outermost shell with only 1 electron, okay? It needs 7 more electrons to fill that up. So the tendency for sodium would be to get rid of that extra electron instead of trying to fill that shell up, okay? So then it will have a complete outermost shell, right? So sodium has a strong tendency to get rid of that extra electron from the outermost shell. On the other hand, if you look at chlorine, that has got an atomic number of 17, right? So 17 protons in the nucleus and 17 electrons. So again, it's 2 in the first shell, 2 plus 8 plus 7, right? So it has got an outermost shell that is only 1 short of a completely filled outermost shell. So chlorine will have a very strong tendency to get one extra electron, right, and fill that up. So sodium and chlorine, it's as though they are made for each other, right? Because sodium has the tendency to get rid of that extra electron. Chlorine has the tendency to accept that extra electron, right? And so the sodium and the chlorine atoms will react, right? And sodium will lose that electron. It will form a positively charged sodium ion because it's lost one negative charge, right? And chlorine will get that extra electron and form the negatively charged chloride ion, okay? So the positively charged sodium ion and the negatively charged chloride ion, they will naturally attract each other, right? And they'll be held together by the bonds of electrostatic attraction, okay? So this is an example of what is called an electrovalent bond, okay? Where there is a transfer of electrons to produce the bond, okay? So next, so this is, further detail on the electrovalent or ionic bonds, right? And so you have sodium ion and chloride ion that are held together by the forces of electrostatic attraction, right? Very strong bonds. So instead of one atom donating an electron, another atom accepting it, right? You can also have the possibility that two atoms could share pairs of electrons, right? And complete the outermost shell, right? So atoms can also achieve completely filled outermost shells by sharing pairs of electrons. And the resulting bonds are called covalent bonds, right? Where the electrons are shared, they're not transferred. So you don't have like ionic bonds, you don't have a positively charged ion and a negatively charged ion, but you have a completely filled outermost shell. So some examples of that, for example, two hydrogen atoms. So hydrogen has got atomic number of 1. So it has got only 1 electron in its outermost shell, right? So two hydrogen atoms could share a pair of electrons between them and they'll form a hydrogen molecule, right? A carbon has got an atomic number of 6. So the electronic distribution is 2 plus 4, right? So it has 4 electrons. It needs another 4 electrons to complete the outermost shell. So what it can do is it can share. There are many different possibilities, but if you have 4 hydrogen atoms, all right, so each one of them could be sharing one pair of electrons, all right, with the carbon atom, and you could have the molecule that is called methane, CH4, okay, which is a gas and a hydrocarbon. Then you look at oxygen. Oxygen has got an atomic number of 8, right? So it's 2 plus 6 is the electronic distribution, right? So it can share, so it needs to really share two more pairs of electrons, all right? So it can share 2 of its outermost electrons with different atoms, but with 2 hydrogen atoms forming water or H2O, okay? Now, the important thing to note is that when you have covalent bonds, see, I said that in the case of electrovalent bonds, those are ionic bonds. You have strong charges, okay? positively charged sodium ion, negatively charged chloride ion, right? But in the case of covalent bonds, okay, they really don't have charges, okay, because you do not have any electron transfer, okay, but the different molecules might have different affinities for electrons, like for, sorry, not molecules, different atoms, all right? For example, hydrogen has got a lower affinity for electrons than oxygen or nitrogen. So if you have a hydrogen-oxygen bond, okay, covalent bond, right, the oxygen will try to pull the electrons much more towards itself than hydrogen. As a result, the oxygen atom will acquire a slight negative charge, right? Hydrogen atom a slight positive charge, okay? So there will be some charge distribution. Charges are very small, but averaged across thousands of molecules, this can be a pretty big effect, right? And it's very, very important in biological systems, okay, for functioning of biological systems. So I will get to that in a few minutes, all right? But before that, let's just define the valency, which is the number of electrons that an atom of an element must donate, accept, or share to form a complete outermost electronic shell, okay? And I mean, there could be atoms that have different valencies also, okay? Like nitrogen and phosphorus, right? Because there are different possibilities. But anyway, hydrogen, so based on this definition, hydrogen has got a valency of 1, okay? Because it shares one pair of electrons with other atoms. Carbon has got a valency of 4. Oxygen has a valency of 2. Sodium and chlorine each has a valency of 1 based on the electrovalent bond that they form. And each carbon can share up to 4 pairs of electrons with other atoms. So now I want to talk about single and double bonds, okay? The single bond means only one pair of electrons is shared, okay? So a single bond is a covalent bond where only one pair of electrons is shared. For example, ethane, C2H6, right? So you have two carbon atoms, all right? They share only one pair of electrons, okay? The carbon atoms are getting the remaining pair of electrons from the hydrogen atoms, right? So this is an example of a single bond, okay? Only one pair of electrons is shared. Double bond would be formed when there are two pairs of electrons that are shared, right? So for example, if you look at the molecule that is called ethene, C2H4, right? So you have these two carbons that are sharing two pairs of electrons, right? And then the rest of the electrons are being shared with hydrogen, okay? Now, for organic chemistry or in biological systems, all right, or in living organisms, this is very important. The difference between single bond and double bond is very important. Because if you look at that single bond at the top, okay, in ethane, right, look at the two carbon atoms, okay. I can freely rotate, okay, one carbon atom relative to the other, right, along that axis. You know, there is nothing that hinders me, right, hinders the rotation, right, okay. Whereas if you look at this double bond here, okay, if I keep the carbon atom on the left fixed, okay, and then I rotate the carbon atom on the right, okay, you can see that the bonds will be crossing each other, right? Double bonds will be crossing each other, right? So I don't have complete freedom, okay? So this is referred to as steric hindrance, okay? And that plays an important role in determining the shape of biological molecules, okay? Because the fact that you cannot rotate it as you wish, right? There's not complete flexibility means it'll put some constraints on the shape of that molecule. And in molecular biology, shape is everything. If you don't have the right shape, you can have catastrophic loss of function, right? The molecules recognize each other from their shape. You know, just like we recognize each other by looking at your face, right? They recognize each other from their shape, right? So if two molecules are supposed to come together and have a reaction, and if one of them, the shape change a little bit, that's a problem. All right, any questions? Okay, so now, as I said before, right, if you in a covalent bond between different elements, the two atoms are going to attract the shared electrons to different degrees, all right? Oxygen has high affinity for electrons, nitrogen has high affinity, all right? Carbon and hydrogen don't have high affinity for electrons, all right? So oxygen and nitrogen atoms attract electrons quite strongly, while hydrogen attracts electrons quite weakly. Therefore, in a hydrogen-oxygen covalent bond, the hydrogen atom will acquire a positive charge, right? Slight positive charge, while the oxygen atom will acquire a negative charge. So overall, the molecule will have an uneven charge distribution, and that's called a polar molecule, right? So water will be something that is like polar because, you know, hydrogen is, the hydrogen atoms in water, they'll have slight positive charge, right? Oxygen atoms will have slight negative charge. Not only that, one water molecule will bond to another one because Hydrogen atom on this is positively charged, oxygen atoms on this are a little bit negatively charged. So the oxygen atom on this will bond with another hydrogen atom of another water molecule, okay, because the positive and negative charges, they will attract each other, okay. And this is called a hydrogen bond. It's a weak bond, but, and that is the reason why water is a liquid at room temperature, okay. It has got very high specific heat, right? Because without that, life would not be possible on this planet, okay? Because the plants, right now it's cooled down a little bit, but you see in this temperature, okay, how will the plants survive, okay? Because they have water, okay? And because water has a very high specific heat, so the temperature is not going to go up that much, okay? And the plants have different mechanisms of dealing with the heat, right? So the overall molecule has an uneven charge distribution, and these are called polar molecules, okay? And I'm getting to the point where you will see why, you have molecules that have a hydrophobic region and a hydrophilic region, right? So because water itself is something that is polar, that has got these hydrogen, positively charged hydrogen, negatively charged oxygen, therefore water will also be attracted to other polar molecules, okay? Because they can interact through electrical charges, right? So the covalent bond between oxygen and hydrogen, the bond between nitrogen and hydrogen, these are all polar bonds, okay? Bond between carbon and hydrogen is non-polar because carbon and hydrogen, both of them, neither of them has much affinity for electrons, okay? So, so that means, see, things that dissolve easily in water, those are going to be the polar molecules, right? Right, because they have that charge distribution. The hydrocarbons will not dissolve in water, okay? Because it's carbon and hydrogen, they don't have any charge distribution, right? So they'll stay away from water. Okay, while oil will stay away from water, okay? So polar covalent bonds are extremely important in biology because it allows molecules to interact through electrical forces, right? And I already mentioned about the hydrogen bonding between water molecules, right? And that's what basically guarantees that water is a liquid at room temperature, right? And it has a very high specific heat, right? It has a high latent heat of vaporization also, okay? So without water, life would not be possible. Okay, and if water didn't have that property, it's just water vapor, would not work. So polar molecules readily dissolve in water, right? Because again, they interact with the hydrogen bonded network in water through electrical forces, right? Non-polar molecules do not dissolve in water, right? So, so molecules which readily dissolve in water, they are called hydrophilic, and they are the polar molecules, right? Non-polar molecules do not dissolve in water, they are called hydrophobic. They hate water, okay? But for biological systems, right, you need molecules which are called amphipathic, right, which have both a hydrophobic and a hydrophilic part, right? That's how you're going to form the cell membranes, okay? I showed you earlier, right? So amphipathic molecules, these are the building blocks for all cell membranes. Any questions so far? Okay, so we continue with our review of organic chemistry. So we are going to look at some chemical bonds and groups that are commonly encountered in biological molecules. Now compounds that contain only carbon and hydrogen, they are called hydrocarbons. Okay, that's a natural name, right? Examples: methane, CH4. Okay, and they have the general formula, right? It's not written on this slide. They have the general formula CN H2N plus 2, right? So if N is 1, you get CH4, right? So that's methane. If you have N equal to 2, you will get C2H6. That's ethane, right? If you have N equal to 3, you will get propane, right? C3H8, right? And then N equal to 4, butane, and so on, okay? So the general structure will look like this, okay? It's C in the center, and then 4 hydrogens linked together by single covalent bonds, right? So that's methane on the left. That's a gas, right? Ethane is another gas, C2H6, right? Now, if you take one hydrogen atom away from methane, all right, you have a very reactive group that is called the methyl group, right? CH3. Similarly, if you take one hydrogen atom away from that ethane, you get an organic group that is called the ethyl group, right?
So when you say they have a general formula, what is they?
These hydrocarbons, right? CnH2n plus 2.
All hydrocarbons have.
At least these gases have. Again, I mean, I probably won't be able to answer that question because I have not delved that deep into this stuff, I don't know, somebody has taken a course on organic chemistry, they could answer that, But,
Alkanes.
huh, what?
Alkanes.
All,
Alkanes, methanes, ethanes.
okay, okay,
So they are specific category of hydrocarbons
okay, okay,
which have single bond with
okay,
carbon.
okay. So you knew the answer, you were trying to get me, that's okay. Right. But I said I was smart that way. In the beginning only I said I don't know everything, okay. So, okay. So methane, ethane, right? And then, and then remember the methyl group, ethyl group, right? So next, we move on to compounds that contain carbon and oxygen. Okay, so those are called. I mean, one set of compounds that contain carbon and oxygen are alcohols. They are characterized by the presence of the hydroxyl group OH, and they have the general formula R bonded with OH. Okay, with a single bond, where R could be any organic group. So for example, if R is CH3, that's one organic group that we saw on the last slide, right? That was the methyl group. Then you're going to have methyl alcohol, or what is referred to as methanol, right? If R is C2H5, then you're going to have ethyl alcohol, which is called ethanol, right? So which is the kind of alcohol that people consume? Okay, what will happen if you consume methanol? Yeah, you're blind, you might even die also. I mean, there's a, huh? Yeah, it's really bad. And there's a gentleman here. He's actually in the mid 70s. Okay, I know him on a personal level. He used to work in the College of Agriculture, and he told me that there was one lab assistant, right, in his lab, or in the lab that he used to work in. That guy, he couldn't stay away from alcohol, right? And he had methanol one day. He was hospitalized and all that. So yeah, all right. Any questions? Then another group of compounds that contain carbon and oxygen, these are aldehydes. All right, aldehydes are characterized by the general formula R. All right, and then you have a group that is C double bonded with O bonded with H. So COH is the group that characterizes an aldehyde. Okay, and R can be anything. If R is H, all right, just hydrogen atom, you get formaldehyde. Okay, and when R is CH3, you get acetaldehyde. Right. Now, see this ethyl alcohol, okay, when that's consumed, right, there is something called alcohol dehydrogenase that will convert that into acetaldehyde, right? And that acetaldehyde is very toxic for your liver, right? So that's the reason why, drinking too much is going to mess up the liver. And the reverse reaction also where alcohol is made, okay, when you do fermentation, right, then, and I will explain those pathways later when I cover the plant biology portion, okay. From sugar, you basically go to acetaldehyde, and then there is alcohol dehydrogenase who's going to give you ethyl alcohol, right? I mean, so one guy in this class knows about it, so he's nodding his head, so I'm saying the right thing, okay. So, okay, any questions? Then another group of organic compounds that contain carbon and oxygen, these are called ketones. They are characterized by the general formula. So you'll have C double bonded with O, that's the carbonyl group, and then it can be linked to different organic groups like R1, R2, right? And carbonyl group C double bonded with O is present in both aldehydes and ketones. An example of a ketone would be acetone. Okay, that's a ketone. Another group of organic compounds are carboxylic acids, right, or organic acids. They are characterized by the general formula C double bonded with O and then OH, and then you can have any other organic group there, right? So this C double bonded with O and then OH, that's called the carboxyl group, right? And R can be anything, like if R is H, if it's HCOOH, then you get formic acid, okay? That's the kind of acid that you have in those hot peppers, right? That's an acid. When R is CH3, right, then it's acetic acid. That's the organic acid that you have in vinegar. Now, what is an acid? Again, from your knowledge of inorganic chemistry, you know that acids are substances that release hydrogen ions into solution, right? So, for example, hydrochloric acid is a very strong acid because if you dissolve it in water, right, it'll dissociate almost completely into hydrogen ions and chloride ions. So it increases the concentration of hydrogen ions. Okay. Same thing with sulfuric acid. Now, the organic acids that we are talking about, these are weak acids, right? Because they only partially dissociate in solution, right? So if you look at an organic acid like shown on the left, C double bonded with OOH, right? It will only partially dissociate. So the reaction goes both ways. It will produce hydrogen ions, right? But it's a weak acid. Right? So because of this partial dissociation, organic acids are called weak acids as opposed to say hydrochloric acid, which is a strong acid, right? Same thing with sulfuric acid, that's a strong acid. Now, we said that, acids are characterized by an increased hydrogen ion concentration, right? So the hydrogen ion concentration is measured using pH, right? The hydrogen ion concentration of a solution, and pH is given by the formula minus log to the base 10 of the hydrogen ion concentration expressed in moles per liter. So if you're looking at pure water, okay, that has got a hydrogen ion concentration of 10 to the power of minus 7 moles per liter. So you plug it into this formula, you get the pH of 7. Okay, that's pure water. If it's acids, right, the hydrogen ion concentration will be higher, right? So the pH will be less than 7, right? If it's a base, the hydrogen ion concentration will be lower, and so the pH will be greater than 7. Okay, any questions so far? So bases are defined as substances that reduce the number of hydrogen ions in aqueous solution. For example, sodium hydroxide, right, is a base because it dissociates in solution to give you positively charged sodium ions and negatively charged hydroxyl ions. But the hydroxyl ions will react with some of the hydrogen ions in water, so it will reduce the concentration of the hydrogen ions in water, and therefore this is a base. Similarly, the gas ammonia, that is a base because ammonia is also going to take up hydrogen ions from water to produce the ammonium ion. Okay, so it's reducing the hydrogen ion concentration in solution. And why did we pick on ammonia? Because again, in biological molecules that occurs. Okay, that's the reason. Now we know from inorganic chemistry that acids react with bases to give you salt and water, right? You know that, right? I mean, for example, sodium hydroxide reacts with hydrochloric acid to give you the common salt, sodium chloride, right? And water, right? So in organic chemistry also you have similar reactions, okay? So instead of a base, you will now have an alcohol because the alcohol had the hydroxyl group, right? OH, right? And instead of an inorganic acid like hydrochloric acid, you're going to have an organic acid. So in organic chemistry, when a carboxylic acid reacts with an alcohol, the reaction is going to produce water, and it's going to produce a compound that is called an ester, right? Which is like an organic salt. You can think of it as an organic salt. Okay. So again, all of this stuff that I'm covering, okay, there's nothing to it other than understanding it, and then somehow it has to get memorized. Okay. I'm not forcing you to memorize this. Understand it and then memorize it, so that you can take an exam on that. Okay. I mean, we're not even assigning homeworks or anything. So at home you should be studying. Next, we move on to compounds that contain carbon and nitrogen, right? And one set of such compounds are what are called amines. Amines are organic compounds that are characterized by the presence of the amino group, right? So ammonia is NH3, right? You take away the hydrogen atom from ammonia, right? You have the amino group, NH2. That can react with some other organic group, like let's say R, it could be methyl group, ethyl, whatever, okay? So R, NH2, right? That's an amine, okay? Now an amine is a weak base, okay? Why? Because it takes up hydrogen ions in solution. It's weak because it's a reversible reaction, right? It goes both ways. It'll take up hydrogen ions in solution, so it's decreasing the concentration of hydrogen ions in solution. Now an amine can react with a carboxylic acid. And amine again, it's like a base. It's a weak base, okay? Carboxylic acid as an acid, so the two can react, right? And they will produce an ester, which is called an amide, right? So you have a carboxylic acid on the left that is characterized by the presence of C double bonded with OOH, right? And you have an amine on the right, which is characterized by the presence of the amino group, NH2, right? The two can react. A water molecule is eliminated, and you have this other molecule that is formed. That's called an amide, okay? This is how proteins are formed. A protein is made up of units that are called amino acids. Okay, so one side is amino group, one side is a carboxylic acid group, right? And many amino acids can be linked together by this kind of linkage, right? This kind of amide linkage to produce a protein. All right, any questions so far?
Said
Go ahead.
formic acid is found in peppers.
In chilies?
Is it naturally occurring or?
I think it's naturally occurring if you find it in chili. I mean,
I
it
know
can.
formic acid is found in ants because that's how they do it.
Yes, 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 yeah. Yeah, so that's another example of naturally occurring, yeah. Yeah, when they bite. At least I was told that when I took, chemistry in high school. Okay, so now that was, for more than 40 years ago, all right? So I don't know if things have changed, but I
Yeah,
was,
I'm searching in Google. I couldn't find it.
you couldn't find it? Okay,
Formic acid.
okay. But what about ants?
Ants is there.
Okay, yeah, then let's take that as the message, Google is the ultimate right now, okay? So whatever they say has to be the gospel truth. But I remember from high school that I was told that it's found in chilies and peppers and all that stuff because those are hot, yeah.
It is used as an antibacterial thing in pickles and all that.
I thought formic acid was always like anthrax.
Yeah, that's what he's saying, okay? But I mean, you're saying that it's then what kind of acid do you have in the case of peppers? That's your assignment, okay? So you find, huh? What?
I think that's capsaicin.
What acid?
Capsaicin. It's like the molecule that makes things spicy.
Okay, I thought it's an. Anyway, I was told in high school, so the fault lies with my high school teacher, okay, who might not even be around right now, but but that's okay. Yeah, I mean, like, let's modify the lecture to say that yeah, it's there in ants, okay. That's good. It's just an example I'm giving, just to, make the presentation a little bit more lucid, okay, instead of just writing down.
If someone asks you, I took a course in genetics and plant genomics, and someone asks, tell me this simple question, where is formic acid found?
No, in ants you can find. That's what you said right now. Yeah. No, no, this is not. So no, this is not a course on plant genomics. Okay, this is a course for engineers, right? So that you acquire the minimum amount of knowledge to become functional in if you have to take a course in plant genomics, the stuff that is 1/3 of this course that I'm going to cover, that is covered in a semester over there. Okay, and the molecular biology also that I covered, the 213 stuff, even without the engineering stuff, that is a whole semester course over there. So if somebody, they will not ask you that. I'll tell you why. Okay, and I, like you won't be asked that question. You'll be asked just the reverse. The other day I was talking to somebody in agri life. Okay, and like in photosynthesis, you have two kinds of plants. They're called C4 and C3. Okay, because like you have carbon dioxide is converted into sugars, all right, in plants. But the enzyme that does that, okay, oxygen is also a substrate for that. So you have photorespiration and all that. And he told me, okay, you're an electrical engineer, you're talking about all that, okay. I said, yeah, I have to learn that because I'm going to teach, all right. So you will not, if you,
But electrical engineers can ask me, that give me this simple
they
answer.
wouldn't even know, okay. They can ask you, but they most probably they would not even know the answer, so, and you can say that I don't know everything, right, so, yeah. Electrical engineers probably will not ask you that, okay, because even the structure of DNA, what I'm covering right now, most of them don't know. In fact,
But it's not formic acid, right?
probably not. It's not used anywhere in electrical engineering.
They might have taken a course in chemistry.
Yeah, yeah, but I mean, like they've forgotten most of the stuff they've forgotten. I can tell you that. I can guarantee you that. Okay, so they've forgotten because if you don't use something, probably you've forgotten. So. Now, for me, the only way I learned these things is because I'm using it here. I'm using it to teach the class and all that. So, but yeah, you don't have to worry about that. In fact, even in Qatar, okay, I was talking to an MD over there and I talked about the MAP kinase pathway and all that. Then he told me, you are electrical engineer. How come you're talking about this? I said, I've learned a little bit about this stuff. So they are, because they are used to electrical engineers knowing nothing about their subject. Okay, so if you know a little bit, okay, about something that they think is not the most obvious thing, they are usually wowed. Okay, so you'll have just the reverse reaction. It's not going to be a got you. You don't know about formic acid. I don't think that's going to happen. I'm not worried about that. Okay, so an amine reacts with carboxylic acid to produce an amide and water. Okay, so So, and this reaction is important because this is the one that usually forms proteins. Okay, we'll talk more about that later. So next I want to move on to phosphates, all right? This is another important group of molecules that you find in living organisms. Phosphates are characterized by the presence of the inorganic phosphate ion, right, which is derived from phosphoric acid, which has the formula H3PO4. So from H3PO4 you take away two hydrogen ions, okay, or two protons, okay, you take them away, you're left with that inorganic phosphate ion. This is very, very important because this is central to how our cells are powered. I'll talk more about this later. So let's just look at a couple of examples, right, of the inorganic phosphate ion. The inorganic phosphate ion can react with an alcohol to produce a phosphate ester, right? I think we're running out of time. Okay, let's stop here for today. Okay.